So, a very warm welcome to all of you guys. Myself, Dr. Nilofer. I'm a bioinformatics scientist at Biotechnica. So, today we are going to have an overview regarding what is meant by NGS, that is next generation sequencing. And as a beginner, which are the important points we all have to remember so that we will get to know more insights about the technique called as the next generation high throughput technique and how it can be used for the research based application. Okay, so before moving ahead, as we all have to understand that what is the need to explore the next generation sequencing. As we all know that the high throughput techniques and the tools where we are able to perform with respect to the identification of novel variants, mutations, or as the SMP kind of mutations is able to identify it using the technique called as the next generation sequencing. Because it is considered as one of the high throughput techniques, will give you a very prescribed findings and even the novel mutations identifications using certain techniques. And that can be even done using the reference genome sequence of your interest. So here, as a beginner, it is need to understand because there is a huge requirement for those kind of students or as the researchers who have skills to know about what exactly next generation sequencing is about. So if you are aware or else if you have to create your skills to understand the NGS technique, so definitely you will get a very good opportunity to perform your analysis and then get a better opportunity for the research context. And moving ahead, for that you all have to know about what exactly the sequencing means first of all, right? So sequencing is carried out for the specific target of our sequence such as DNA, right? Majorly. So here in this next generation sequencing is majorly categorized based on the genomic sequencing. So genomics is nothing but a sequencing is performed or as the analysis of the gene and genome is done. Okay, so that's why we used to perform the analysis of DNA sequence majorly in next generation sequencing approach, because we are able to take help of certain biological sequence in the form of DNA, and then we are able to identify what are the amino acid change that's been happening so that we can target those particular protein targets and then we can look for the identification of potential drug targets. Getting all of them? So using certain methods or the technology, it has enabled us to determine the order of nucleotides, okay, the exact pair of the nucleotide, so that we can identify what is there is in the change or else any kind of mutations has occurred. Got it? So for that particular purpose, we all have to know the very basic overview is nothing but what is DNA sequencing, right? So DNA sequencing is nothing but a figure outing the order of DNA nucleotide on the basis of ATGC pair. So with respect to the reference genome, so that we will get to know the overall makeup of an organism by a reference genome sequence. And that is able to understand by inventing or else identifying the specific method called as the Sanger sequencing, right? And for that method, you all have to know that which are the traditional methods were available and why the NGS technique is playing very significant role in the current high throughput era, right? So traditional methods of sequencing and its limitations first we all have to know. Then only we are able to overcome the challenge and understand the role of next generation sequencing for the research based application. So as we all know that previously we used to have certain different methods are even available, right? So such as maximal hybrid method, which uses the radioactive levels to perform the sequencing, right? So that we will get to know the specific levels within the fragments of DNA, and based on that we can identify the mutations accordingly. But after that, there is a invention of Sanger methods. It's nothing but the Sanger sequencing. So what exactly the Sanger sequencing is to perform, the principle is nothing but it utilizes the fluorescent dye, okay, for labeling the DNA fragments. And once it's done, then the separation of the extended fragment of DNA within the addition of deoxyribonucleotide with respect to the lack of 3-OH groups is formed. And then a chain terminations is occur over here. So basically, it will fluoresce the specific fragments by labeling accordingly, and then we can able to identify the mutations. So it do have certain limitations, such as it is slow and it cost high per run, because it is using ultimately the dye and the radioactive levels accordingly. So to overcome this challenge, we need certain technique which should be very specific, accurate, fast, and even economical benefits, right? So as we all know that for reaching the next generation sequencing, what are the eras of sequencing was there, right? Such as the first generation sequencing, it's still available, which contain a sequence of many identical molecules, right? A similar molecules is able to perform the sequencing over here. Then the sequencing of the large gel or else the capillary tubing limits scales. It's nothing but we used to take help of the samples and then we used to perform based on the capillary tube or else the gel format, right? So that was the first generation or the era. We used to take the help of Illumina amide sequence technique. Got it? So this is exactly how the experimental look like, or as the experiment of the specific technique has available. Then we have a next generation with respect to the second generation sequencing before that. Second generation sequencing is nothing but where we are able to perform the sequencing of millions of clone amplified molecules per run. Got it? And it is pretty good. And then we are able to perform even using the reversible stepwise sequencing and immobilized surface method. Okay, so these are the some 2 to 3 additional methods we used to perform based on the sequence. So these methods were available based on the experimental techniques with respect to the era of sequencing. Okay, so we all have to know how the era of sequencing was available, and using the next generation sequencing, what kind of findings we are able to achieve, right? So as you all can see that discussing with respect to the next generation sequencing technique. So next generation is nothing but the, as I mentioned previously, that it is one of the approach of the DNA sequencing, such as the genomics data sequencing. So it's called as one of the high throughput DNA sequencing technique. So why high throughput? Because we are going to discuss ahead. So it employs the micro and nanotechnology because it reduces the sample size. It uses the low cost reagent and also the less time to get the results accordingly. We can even perform the massive parallel sequencing and even the sequencing of thousands of sequences at once, right? And it will even produce an enormous amount of data. It's nothing but whenever you can see that the next generation sequencing data will be available in the form of GBs file, right? Maybe it's a 1 GB, 4 GB, 5 GB, 20 GB of data is available, right? So because we are able to perform the sequencing of the samples of the thousands of sequencing samples at one, right? And it can even perform the massive parallel sequencing at a time. So that's why we are able to get the results in the huge amount of data, or as the big data needs to call us in today's era. Got it? So why it is possible? Because it's using the macro and nanotechnology as a technique, and even it is using the high throughput DNA analysis. The one of the limitations which is get overcome over here is that it reduces the sample size. Reagents are used comparatively are low of the cost, and even it takes less time to give the results accordingly. Got it? So what exactly then the next generation sequencing is about? Yes, so we all have to know that. So next generation sequencing is called as the automated Sanger method first generation. Okay, that is the first method was there. But technology is developed after that is known as the next generation. Okay, because that was the first generation, as we have discussed in our previous slide. After that, whatever the accurate and quick sequencing technique was available, that's what is called as the next generation sequencing. So basically, NGS has enables the sequencing of biological code at a very rapid pace and with the lowest cost per operation. Okay, that is one of the good significance of using next generation sequencing. So this is a primary advantage to overcome the conventional methods. For example, billions of short reads of can be sequenced in a one operation. So it's nothing but the fragments, right? What you are interested for. So that can be sequenced with respect to the in a one set of operation. Billions of short reads can be sequenced. So that is one of the very great application of next generation sequencing. Because majorly the platform of next generation sequencing use the Illumina. Okay, so Solexa is the one of the technique that is called as the Illumina, and Solid, that is by applied biosystem, and 454 by Roche is been. These are the major platforms used in the next generation sequencing, and that's why the techniques or else the analysis we used to do very fast, accurate, and with a less period of time, and millions of short reads can be performed with the sequencing method. So what exactly the next generation sequencing consists of? The next generation sequencing consists of the sequencing of the combination of strategies. Combination of strategies are nothing but first it creates the template that is called as a template preparation. Then it will look for the sequencing and imaging so that you will get to know the exact fragments with respect to your targets. Then afterwards it will look for to perform the genome alignment. So that is one of the most important steps which is included in the next generation, and that's why we are getting very accurate results with respect to the reference genome sequence alignment is done, right? And then finally, once it's done, then it will also move to assemble your methods. It's nothing but to identify the variations with respect to the known or as the novel, and then again it will assemble in a such a way that we will get the findings with respect to the outputs. Getting all of it? So a quick overview that Sanger's versus next generation. So Sanger sequencing with respect to the sequencing sample, it can be cloned or it use the PCR technique. Whereas in next generation uses the DNA libraries to make the sequencing of the sample. Whereas the preparation of the steps, it is a few, right? And use the reactions to clean up, such as the fluorescence accordingly. But here you can work with a complex procedure to perform the NGS steps, right? Then we have the data collection. So you can collect the data with respect to the samples. But here you can collect the samples with the slides, right? Whereas the data, it's read per sample. That is the one of the limitations or the drawback of next generation sequencing, why it is included, because the Sanger sequencing used to take one read per sample, right? Whereas the next generation sequencing is taking millions or is the thousands of reads per samples. Okay, and that is one of the great application of using next generation sequencing for the analysis and even to look for the accurate and quick results accordingly. So if you just go walk through the particular next generation workflow, so you can see that it creates a DNA fragments. Okay, so this is a DNA, got it? And after that, it will create the fragments. Okay, so these are the fragments of DNA. And once the fragments of DNA is then it will add the platform specific adapter for the sequence to every fragments. Okay, it will take and read the every fragments and it will perform the specific adapter sequence. So this specific adapter sequence are nothing but it will look towards the molecules, okay, which is in the binding format. And once the adapter molecules bound to the DNA, then those adapter molecules are nothing but the bind libraries to flow the cell or the beads, right? So add the sequence primer. So basically it's a primer, guys, remember. Or as the binding side that adds the barcodes for multiplexing with respect to your target of your sequence samples, what you are interested. Getting all of you? So first step is create a DNA fragment, and the second step is to add the platform specific adapter with the sequence to the every fragments which is used accordingly. Whereas if you just go through the workflow, you can see that this is a really best example. Sample extraction, DNA fragment, and in vitro adapter ligation. So it's nothing but this is the DNA, right? It will just fragment accordingly. and then it will look for the particular DNA fragmentations, right? And then you can even look for the cloning or as the particular PCR technique, or else you can even look for the clone amplification by bridge PCR. Okay, and afterwards you can see here that it basically use the sequencing method, which is called as a pyro sequencing, and the sequencing by the ligation. Okay, that is a platform what we have discussed previously. And then it will even use the Solex, that is the Illumina based technique. So these are the best techniques used to sequence by the synthesis. And then according to the workflow, it will give you an outputs, right? So NGS technology overview is nothing but NGS defers the template preparation, sequencing, and even imaging with the data analysis. So these are the 4 to 5 important steps are available, and that's why we are able to quickly walk through with the NGS data, and then we are able to adapt those techniques, right? So basically, next generation sequencing uses the important steps what we have discussed, and commercial available technologies are nothing but Illumina, Solex, okay, Roche 450, right? Biosciences, then the solid system, Pacific Biosciences, ion tolerant technology. So these are the techniques is used to commercialize the technology, and then it has been used majorly with the startups company or else the major companies who are working with respect to the sample screening. They used to work with the NGS technique. Got it? So as you all can see that the workflow is nothing but in the tube, DNA extraction is done. Whereas in the second step, you can see that there is a step called as a library preparation. So it will prepare your library based on the DNA fragments. Once the adapter is connected, so it will call as a DNA library. Okay. So first DNA, then the fragments will be done, and afterwards the adapter is get added, and finally it is called as a DNA library. Got it? Whereas in third step, you can see that it is called as a sequencing. So using the specific NGS technique, we used to able to perform the sequencing based on the fragments, based on the thousands or the millions of fragments versus the sample, right? So we will get to know the specific nucleotide expression patterns, and that data will be saved in the form of FASTA-Q file format. So FASTA-Q is called as a quality control. So it will give you the quality of your every nucleotide accordingly, right? So after that, what we used to do was that we can take this FASTA-Q file, and from this step, using the bioinformatics approach, we can look for the align the reads with the BAM file format, and then finally we can identify the variants using the VCF file format. So basically, VCF file format is called as a variant calling file. So there is a high chances that in this particular file, you will get a novel variants or as the SNP kind of mutations. Getting all of you? So these are the basic steps or the workflow it will perform with NGS technique. So in the template preparation is a very sensitive, okay, most sensitive step is there for the image technique. It is not able to detect in the single molecule, or as the amplification of the templates is done over here. Okay, so cloning of amplified template is an important step over here. So in that we used to take the PCR, EM PCR, or as the 454 solid technique is being used, such as Illumina, right? And then finally, we used to get, once the cloning is done, so we used to look for the single molecule templates. Okay, once the single molecule templates is available, then we used to move for the immobilization. It's nothing but if you just make a quick comparison of traditional versus NGS, then you can see that immobilization of the template fragment over the bits or as the glass bits allows the billions of the sequencing reactions to run simultaneously. So that is exactly what we need in the template preparation. And then genome alignment and assembly. Again, that is important because after the NGS reads have been generated, so they are aligned to either to a known reference sequence or as the assembled to the de novo. So reference genome such as the human reference genome 38, okay, or is the 37th, or is the human reference genome 19 projects, right? Or is the de novo sequencing will done. Got it? So this step is again important, that is called as a genome alignment or the assembly. So as you all can see that basically the next generation sequencing is used by the researchers in three main approaches. The first is genome sequencing. Okay. So if anybody is planning to get the next generation sequencing to get adapt, so they used to take genome sequencing. It's nothing but they are interested to perform a whole genome sequence analysis. Okay. Based on the DNA and then the fragments and then finally get the reads. Okay, maybe 1,000 reads or the billions of reads at a time. Got it? The second one approach is called as the exome sequencing. Exome sequencing, it's look to the exomes and based on that, it will fragments and then it will generate the reads. Got it? So that is one other approach. And the third important approach is also available, which is called as a targeted gene panel sequencing. So basically here you can target the specific genes rather than targeting the entire genome or as the exome part. You can look for the targeted genes and then you can fragment those things and then look for the reads generation. And then accordingly use the NGS pipeline to get the VCF files. It's nothing but the variant files accordingly. Once the FASTA-Q file is available with you. Got it? So as you all can see that major after you get the FASTA-Q file and that FASTA-Q file will be available in the databases of bioinformatics platform, right? And from there we used to download the FASTA-Q file format and then using the command line probe in the bioinformatics platform, we used to process the FASTA-Q file in the standard pipeline and then followed with that one, we used to get the BAM file and the VCF. And finally, in the VCF file, as we have discussed earlier, that we will get the different variations information with our target of our interest. So if you just see that why we used to do such a huge analysis is nothing but it do contain some applications. So NGS is used with the drug target identification. And NGS technique will also help you to identify the personalized target therapy and then identify the potential biomarkers for your interest. You can even use for the biopharmaceutical application. And this technique is even playing a very significant role with the virological study because the overall sequence analysis based on the fragment generations and then genome alignment with respect to the reference genomes can be done so that we will get to know the similarity and even the evolutionary information about the targets, right? So that's why the virological study or the pharmacogenomics approach is also included with NGS. So pharmacogenomics is nothing but where we used to study the drug with integrations with the genomics data. So that in future we can even come up with the findings of personalized cancer therapy or is the personalized drug targets, right? So then the next important application is the polypharmacology. It's nothing but where we can look for the huge number of drug targets once the sequencing has done properly, right? Then the epidemiology is nothing but where we can look for the specific target of our interest to sequence the particular techniques and then look for the specific approaches. So as you all can see that a quick overview with respect to the advantages of next generation sequencing is nothing but the over here, right? So the Sankar sequencing and the next generation sequencing. So we have discussed the both, right? So looking towards that, you can see that the sequencing of the advantages with next generation is the low reagent and the cost, reduce the sample size, less time will be taken. And compared to that, you can perform the high degree parallelism with the capillary sequencing. And then you can even look for the cloning, right? Picking the transformations or as the non-in vivo cloning can be even done. So that is the best things to go through and then look for the applications accordingly. And that's why these applications are really good. So that's why it is getting adapted with many commercialized company, and even the researchers are adapting this so that they can identify the potential findings. So quick applications of NGS technique, as you all can see that it is used for the mutation discovery, right? To get to know and study the transcriptomics analysis using the RNA sequence data. You can even work with the sequencing of the clinical isolated strains with the reference mechanism, right? It will also enable us to study the metagenomic study, right? So metagenomics is nothing but where you can look for the samples from the soils or the human gut, and then you can find the new species targets of your interest, right? You can even look for the DNA and protein interactions with the chip sequencing technique. So NGS technique will also help you to discovering the non-coding and RNA sequences accordingly. It will also help us to discover the molecular diagnostic for the oncology or as the inherited disease of your targets. So NGS technique also help us to give you a gene regulation analysis, whole genome sequencing, and eval exploring the chromatin packaging of your targets. So that is a great application of next generation sequencing, and that's why you can see that in every research point of view, genomics next generation sequencing is playing very significant role. Overall summary is that next generation sequencing has changed the way to carry out the molecular biology and even the genomic study, right? So it also allows us to sequence and even annotate the genome at the faster rate. For example, now you think that I'm interested to work for the next generation sequencing, so you can perform the analysis based on the integration of the in vitro and in silico study, right? Because we have a standard pipelines, we have a databases in bioinformatics platform where it will help you to process your FASTA-Q file to identify the variant calling files accordingly, right? So it will also allow us to study the variations, expression patterns, and even DNA binding at the genome-wide level study. Okay, so that is a quick summary. I hope that all of you have understand this. Okay, if anybody have any queries, any questions, definitely they can drop your questions accordingly, and then we will revert you regarding the same. And you can see that if you're really interested to explore more about the overall next generation pipeline, or else you are interested or willing to land up with any kind of projects, so we biotechnicians are here to help you guys regarding guiding you with respect to the any kind of bioinformatics related projects or as the omics related projects. Because as you all can see that in this overview, we have given you only the introduction, right? So we have certain databases in bioinformatics platform where it will help you to design the overall pipeline. For more references, what I can highly recommend you to all of you guys is that just kindly read certain reference papers, okay? It will really give you a good overview that how exactly you have to understand the approach of next generation sequencing, and then you can even adapt for your own research perspective, right? So that is all about, okay? So if you think to enroll more with the projects, definitely you can. Thank you guys. Thank you very much.